The study that we recently published is describing a new approach for enabling targeted immunotherapies for acute myeloid leukemia and other hematological malignancies. And in particular, it's focused on epitope editing. So it's the precise modification of targeted epitopes in normal hematopoietic stem progenitor cells. And this enables these cells to avoid direct killing by CAR T cells or monoclonal antibodies. So it's a way to make them resistant to targeted immunotherapies. And in this way, we can effectively create an leukemia-specific target, and we can improve both the safety and the efficacy of these adoptive immunotherapies. What we did in our study was start from receptors that were expressed at high levels on the leukemia cells, and so they're very well-known targets for targeted immunotherapies. By epitope mapping and library screenings, we identified a single point mutation that could abrogate the binding of therapeutic antibodies and CAR T cells. And then we introduced these mutations into primary CD34+ HSPCs by means of base editing, which is a novel and very interesting technology which avoids the toxicity of conventional CRISPR-Cas nucleases. And then we validated that these mutations do not affect the functionality of these receptors. So we did not affect their capacity to bind the ligands and to accept their function. The modified HSPCs are fully functional, but at the same time, they become resistant to the targeted immunotherapy. And we validated this through in vitro screenings and also in vivo experiments. We were able to confirm that the CAR T cells that we administered were able to eliminate selectively just the leukemia cells while they were sparing the normal epitope-edited hematopoiesis. When we started screening for mutation that could abrogate the binding of the therapeutic antibodies and CAR T cells, we identified, to our surprise, single point mutation that were able to disrupt the binding of the antibodies. And after that, we were able to introduce the same mutation through base editing. And in particular, we were able to identify guide RNAs and editor variants that could introduce these mutations into primary cells with adenine base editing for all three of our targets. This is actually one of the key results because it allows us to multiplex editing strategies. So we can insert more than one epitope change at the same time. And this allows also the use of multi-target immunotherapies. Again, this which has an undeniable advantage compared to therapies that target just one surface antigen. After that, we moved to the validation of the functionality of the receptor. And again, when we screen the mutation that we identified in our experiments were able to reproduce the receptor functionality, we found no difference compared to the wild-type receptors. And also epitope-edited primary HSPCs were able to preserve full stem cell functionality and in vivo repopulating capacity. When we moved in vivo, we wanted to confirm that our epitope-edited hematopoiesis was resistant to the targeted immunotherapies. We exploited an advanced in vivo model with co-engraftment of both healthy hematopoietic cells and patient-derived AML xenograft. And we then treated these mice with FLT3 and CD123 CAR T cells. And in both cases, we were able to confirm the possibility to eradicate the leukemia cells and at the same time, the epitope-edited hematopoiesis, so the healthy cells derived from our modified HSPCs, was preserved, was resistant to them, and was not depleted in the same way as the ABS1 edited controls. And this, again, is the key finding because we are effectively eliminating or greatly reducing the on-target off-tumor toxicity of these immunotherapies. So I believe that epitope editing is a really promising new technology that can definitely be translated to clinical practice. Very importantly, we are exploiting an allogeneic stem cell transplantation platform, which is the standard treatment for high risk or relapsed AML patients. So we are adding on top of what is the current standard of care, the possibility to exploit a very potent targeted immunotherapy, be it CAR T cells or antibodies or bispecific antibodies, that can allow the patients to possibly eradicate the minimal residual disease after allotransplantation. The outlook is that high-risk patients or patients that do not fare well with the first lines of chemotherapy could receive an edited transplant, and after the transplant, they have the possibility to receive a very potent and effective CAR T cell therapy without having the risk of on-target toxicity. And this allows therapy to act for longer. We can imagine this even as post-transplant maintenance that can protect the patient against relapse in the long term after allotransplantation, which again is the same therapy they are currently receiving nowadays. And secondly, epitope editing can also be exploited in the context of non-genotoxic conditioning. This is a new approach to hematopoietic stem cell transplantation, which avoids the toxicity of chemo and radiotherapy, which is normally used for myeloablation before transplantation. It's required to clear the bone marrow niche to allow stem cell to engraft. But in this case, instead of using chemo and radiotherapy, you're just using an antibody targeted at stem cell antigens. The problem with this approach is that, again, you risk eliminating the same cells you want to engraft. So the cell product that administered may be depleted by the same conditioning agent because of the pharmacokinetics of these antibodies. And again, epitope editing would enable these cells to become resistant to the conditioning itself. And you can imagine using this as an in vivo selection. So you could theoretically reach higher chimerism and higher therapeutic thresholds, even when you by administering the antibody after the transplantation. So modifying the cell products used for gene therapy or even allotransplant and using a non-genotoxic conditioning is another very important application of epitope editing.